So introducing now Dr. Timothy Button. He is from New Zealand. He was born and bred there, as he put it, and he's a statistician with expertise in genetics, statistical genetics, and research and industry has been his battlefield for 6 years now or more, counting. And so he's very used to it, and he will share his experience. So I'll go through into the detail of his bio because he's going through that in his talk, but he has a lot of experience in that area of research and also coordinating these two kinds of environments. And he's also been a representative of the Student and Early Career Network in the New Zealand Association for Otago. And most importantly, as we heard from G in the last talk, he has the heart and the skills to help students and early career statisticians. So we're very pleased to have him here, and we're looking forward for this great talk, which title is The Academia and Industry War, Surviving the Conflict Between Two Opposing Ideals as a Graduate Student. So thank you very much, team. It's a pleasure to have you here.
Cool, thank you, Danielle.
Take the floor.
I'll just share my screen. All right, hopefully you can see that.
Yes,
thank
you.
All right, thank you everyone. Thank you for coming along today. I just have to admit, the title I wrote for this talk is probably somewhat controversial, if not provocative, but it was intended to try and get an audience. So I'm quite glad that we're getting reasonably good numbers along today. But I have to just start by first making a few disclaimers. So let's see if it's gonna. Yeah, so the first disclaimer I want to point out is that what I'm going to talk about today is going to be in very general terms. If we're talking about averages, it'll be thinking in terms of that, but as statisticians, we know there's often outliers and exceptions to cases. So just bear in mind, this is general terms and not specifics. The second thing I want to just disclose is that I come from New Zealand. I've worked mostly in New Zealand. So what I'm talking about will be mainly in reference to the New Zealand context. I've had a little bit of experience in Australia, but not a lot really. The other thing I want to point out is I'll be talking about this from a statistical sort of expertise. I know there's other disciplines where things are probably a little bit different. And last year, after putting my title in and my abstract, and when I started putting this presentation together, I actually thought, well, actually, maybe my title is a little bit misleading, and just let me explain why I think that is. So if I've put this graph together to kind of illustrate this point. So here on the x-axis, essentially those who you're working with. So we've got on the far left the clients, the customers, on the right the general public, and on the y-axis we essentially got what the ultimate goal is. So I've got the top here, public good, and the bottom sort of commercializations. And the way I kind of see it is, at the top you've got the universities, which are more or less out there for public good and for the general public. Though I mean, again, this is generalizations, there are always exceptions. And I know that a lot of universities there's commercialization and things like that. And then down the bottom corner is essentially your multinational corporations or your companies. They're very much on making money, producing products and serving their customers. And in the middle, I've put what I've called research institutes, or particularly Crown Research Institutes. So these are the government funded ones. And in general, they kind of sit in the medium between the two. And so the way we can see it is up here at the top, it's all focused around kind of publications, a lot of freedom of research, fund the research from teaching and grants and things like that. Whereas down the bottom, we're worried about commercialization, and it's often hard to really publish your research due to IP restrictions and things like that. So where I sit, or what I'm going to cover today, is actually between essentially these two here. So sort of the university's academic part and being in a Crown Research Institute. So I kind of feel like my title's a little bit misleading because I'm not truly in industry. down the bottom left here, where, yeah, it's really hard to publish your research. So that's just my starting disclosure. So I thought I'd also begin by just giving a bit of background of myself so that people get an idea of where I'm coming from. So I actually grew up in Auckland, a place called Stanwell Bay, and I spent the majority of my childhood there. And then when I was about 15 and a half or so, moved down to Palmerston North, finished my high school years, and then went and studied at Massey University. Did a Bachelor of Science in Statistics across 4 years, so yeah, working there. And as part of that, I think in my second year, I ended up doing a summer project with Professor Martin Hazelton, looking at sort of polytope samplers. And then in my third year, while I was still studying at Massey, I managed to get a summer scholarship at the Australian National University, where this was with Professor Alan Welsh. Again, this was just for a summer, so it was an 8-week project, I think it was, and I was working on sort of the lasso selection for random effects in linear mixed model contexts. I then came back, finished off my degree, graduated, finished in 2015, graduated the following year. And at the time was applying for various jobs and ended up getting an internship down in Ag Research in essentially what we call Mosgiel, which is just outside of Dunedin. And not quite sure what I wanted to do after I finished my studies. I, was offered to do a PhD. It kind of came out of the internship that I was doing there. And so I wasn't quite sure whether I wanted to do a PhD. This might sound a bit strange to some people, but I was a good student, I had good grades, and I had a lot of professors trying to convince me to do a PhD. Nothing really appealed to me at Massey. I even went and visited Auckland University and talked to lecturers there at one point. But again, what I really wanted was something that was very applied. And so the opportunity to do a PhD with Ag Research was quite appealing. So while I was trying to sort that out, I went back to Palmerston North, which was home at the time, and I managed to do sort of 3 or 4 months work for Ag Research up in the Palmerston North campus. Pretty much a prelude to my PhD, getting some experience doing real research on site, and then came back down to Dunedin, where I started my PhD in the end of 2016. And after deciding, or juggling around about which university I'd go with, I decided to just go with the one that was in town, which was the University of Otago. So that's kind of roughly my background and how I ended up where I did with my PhD. And I just want to talk roughly about my PhD experience and some of the things that I've learned along the way. So just to kind of give a highlight or an overview of how my PhD was structured. So I had, from the university, I had 2 supervisors. So Associate Professor Matt Schofield was my main supervisor, and Professor Mac Black was the second university supervisor. And then I had an advisor with Phil Wilcox, who were all in the Department of Mathematics and Statistics at the University of Otago. And then I had a supervisor from Ag Research, Dr. Ken Dodds. He was pretty much my mentor, and he was also my summer intern supervisor. And then while I'm at Ag Research, I was involved in various teams, mainly in the bioinformatics and statistics team, but worked quite closely with animal genomics and forage genetics. Now, the interesting thing was I ended up getting a scholarship with the University of Otago, so a doctoral scholarship. But I was also had funding from a program that Ag Research had. So the Ministry of Business, Innovation and Employment dish out quite large research programs called Endeavour programs. They're roughly about 5 or 6 years. And we had one called Genomics for Production and Security in a Biological Economy. So I essentially had joint funding. And this is quite a key component of my PhD, because essentially it meant that I had a lot of freedom in terms of how academic I wanted my PhD to be, or how applied I wanted my PhD to be. So Whereas if you're fully funded by, say if I was fully funded by Ag Research, then I would have had to probably do research more aligned to the program, and there probably would have been less scope for more academic type research. So to me, this was quite unique and quite special. It gave me a lot of opportunity to essentially do the kind of research I wanted. Oops, sorry. The other point I would make was pretty much I split my time half and half between the two institutions, so I got a good feel of what it was like being a student in both. Right, so just as another thing I forgot to mention was in this talk, I'm not really going to talk much about science or the statistics. It's going to be much more an overview, sort of a career type presentation. But just briefly with my PhD, I just want to give a background of kind of how it fitted in the context of what we were trying to do. So the program that from Ag Research, it was partially funding my PhD, was focused on developing a sort of high throughput sequencing method for genomic data. And the method was pretty well designed to be very cheap. So we wanted, it was designed to try and get as low cost as possible, and we got down to about $30 New Zealand to sequence one sample. And we also wanted a method that was quick, so we could turn around from when the data was the sample was collected to when it was analysed, a sort of 4 to 6 week turnover. And so there was a big push for this, and this was needed to be able to apply it in industry. We also found the method was quite applicable, so it was applied over, I think for the course of the program, was over 100 species that it was actually used in, so it was very, very general. And there was a lot of other features that made it quite appealing. The problem with the method, the problem with the method is that the data that it generates is actually a lot messier. There's a lot more missingness in the data, so missing data, more errors, which is a real big one. And really, the messiness of the data was as a consequence of how the protocol in the lab was designed. And so you could have argued that, well, why use such a method? But the point is that it is cheap, it is quick, and we can apply it readily across different species. So how do you handle the data was really the question. There's a couple of ways that you could have done it. One was basically to do more sequencing, but that really costs a lot more money and defeats the purpose of what we were trying to do. The second thing that often can occur, and we sometimes see it in research and just literature, is that people who do not understand the data often ignore or don't handle the errors in the data and just use out-of-the-box kind of software. And we found that often when people did that, the results did not make logical sense. It was, and a lot of people thought the data was rubbish because they weren't actually modeling the data or analyzing in an appropriate fashion. So essentially my whole PhD came around developing new statistical methods to handle and model the data. So basically that we could use it on the kind of standard analysis that we used to with genotypic data. So that's kind of the motivation for what I was doing. It was very much tied in with what we were trying to do with the method. Right, so my PhD was structured into 3 parts, essentially. So you could say that each part was what was one paper equivalent. So the first part was very applied. It was essentially trying to take the data that was generated and apply it in specific analysis, which we called linkage mapping. And the model we used in this context was just a hidden Markov model. But to build in sort of model the errors in the data was sort of a novelty of what we did with this first part of the PhD. So it was very applied. We managed to get a paper out within a year. And I actually ended up using quite a few data sets later on. The second part of my PhD, which essentially ended up being an extension of the first part, was some ideas that came about from one of my supervisors who had expertise in hierarchical modeling, and also particularly was quite keen on Bayesian, using Bayesian to fit the models. So that's what we ended up doing. It was quite a lot of fun. I quite enjoyed it. But the point I want to make here is this was probably more the academic part of my PhD instead of being very applied. And in many ways, the work I've done here is probably least likely to be taken up in practice just because of the computationally it's quite expensive. And so, yeah, it was just interesting how I started off very applied. I moved into something much more academic, which the way my PhD was structured, which allowed me to do that quite easily. And then the third part of my PhD was related around developing a new estimator for estimating relatedness to genetic relatedness between individuals. Again, count for the errors in the data. The novelty of this was actually that it was done on polyploid species, and if anyone who's done work on polyploid species will know that the genetics gets very, very complicated very quickly with these species, and so developing appropriate tools is quite hard. Now, there's an interesting story around this third part of PhD, because when we first discussed it amongst my supervision group, there was a lot of skepticism about going down this path, mainly because not wanting to go down a rabbit hole. And I think it kind of reflected a little bit, perhaps the difference between academia and industry in terms of thinking, because in industry, this was something we wanted to do. We didn't know if we could do it, but let's give it a shot. Whereas academia, I suppose, wanting to get papers out, wanting to produce output, a little bit worried about getting stuck in holes. So it was interesting. I ended up doing it, and it turned out to be really successful. But yeah, it was just an interesting part of my PhD. Oh, sorry. And as part of my PhD, I wrote a package just about for every part. And this came about because another one of my supervisors who was quite keen on packages and tidyverse type idea came up with the idea. So I kind of merged them together in kind of like a tidyverse style collection of packages. So in terms of what resources I got as a PhD student, I've kind of listed it a little bit here. In many ways, what I got at the university was pretty similar to what was on Access at AgResearch, at the CRI. There was some little bit of difference, like the journal access is like a lot better at the universities. But in terms of like IT resources, it was fairly similar. You both had a community. It was a different community. The university was more students and academic staff, whereas AgResearch, it was much more general with the biology. But yeah, the two main differences probably are working in a CRI, or being associated with CRI, we generate an awful lot of data. And so there's always plenty of data sets around that we can use for developing methods and looking at. Whereas I've tended to find in academia, universities, that getting hold of data sets can be a bit of a challenge. And then just having that expertise around the scientists, as well as not just the statisticians, was invaluable for this kind of project. And I just want to build on that a little bit. So here, I've just kind of outlined for this particular program, MB program that I was a part of, sort of the structure of our team and how we kind of work together. So sort of on the bottom left here, we have the sort of data collection, and then you move on to your sequencing, and then you proceed to your analysis. And the different sort of expertise that essentially take responsibility for that part of the process. So you've got your field team that are responsible for the data collection, you've got your lab staff for handling the samples in the lab and then getting the sequencing. Here I've used bioinformaticians. I know there's a bit fuzzy between where the line gets drawn between bioinformaticians and statisticians, but I'm thinking in terms of taking the data, mapping it to genome assemblies, processing that data in a form that you can then use to analyze, which often get handed over to our statisticians. And then overseeing this whole process is really the scientist with all the biological questions. And really one of the things that I want to get across here today is, as a statistician working in this kind of environment, you're not just waiting for the data and you're actually working closely with bioinformaticians, with the lab staff, with the scientists, because of what happens down the process ends up affecting your data and affecting you. And so you work close to them to understand what's going on, or whether changes in their processes are needed in order for you to actually get good data. So this is, yeah, really cool part of being a PhD student while you're in a CRO. One thing that I did find, talk about freedoms. In academia, one of the real advantages is you get to choose essentially what kind of research you want to do. The freedom to do it is quite special. The big caveat probably is that whatever you do, you need to publish it, because that's really how you get your funding. And there's a big push to get in high impact journals. And also, the teaching funds the research. So I think not always, if you're in an academic position, you kind of need to do the teaching, and not always, you don't always have same level of control on what courses you like to teach. In industry, things are a little bit different. The research you do is driven by essentially things like the science plan of our company, so that determines what our focus on in terms of our research. You'll have project milestones that need to be delivered on. There's often ideas we have for doing research, but it depends on whether we can get funding for it. And really, there's a big drive to make it practically relevant to industry. And then there's company policies that need to be followed, and there's multiple things to think about here. So you have like travel policies and things like that. But I have got a real funny story, somewhat, well, out of the ordinary. But when I was doing my PhD with the first paper that I was writing, I did a simulation to compare standard software or software out there, existing software, with the new method I developed. And one of the software that I was looking at was a Windows-based program that had no scripting functionality and was point and click only. And so with some help from the IT group here at AgResearch, we wrote a C# script to automate the whole process of the point and clicks to do it. And that worked fine, but it required that we had an unlocked screen the whole time. And apparently that broke company security, the company's security policies and requirements. And to get around this, I was asked to put a lock on my door so that when I was running the simulation, that the door was locked when I wasn't there. So anyway, this kind of reflects a little bit some of the things that I think you occasionally have to deal with when you're in a company or a research institute that I don't think you'd ever have to deal with in a university, but that's me anyway. So just to finish up the PhD side of things, perhaps a bit of advice for PhD students or those on the call who might be thinking about doing a PhD or in the process of applying for one. To me, that there's three key components that you want for a good PhD. First is you need a reliable source of funding. The second one is you need a topic that interests you enough that you can enjoy it, but also be able to spend 3 years researching it. 3 years is quite a long time. And as one of my supervisors kept telling me, this is about the only time in your life where you have the opportunity to spend such dedicated time researching one topic. So enjoy it. And the third component is to have a good supervision team. And I was really blessed to have supervisors that worked together really well, worked well with me. Obviously, they had their particular points of view and the particular bents of research they wanted to do, but at the end of the day, they let me make the decisions about what to do, and that was actually a really, really good team that I had there, and I give them a lot of credit for my success. Couple other things I just want to mention about PhDs, and I think this often gets left out, is PhDs is not just about research. It's about all the skills you develop while you're doing a PhD. So it's, when you're presenting at conferences, there's presentation skills, or there's collaboration skills, or getting your name out there are really important. So the coding skills you develop along the way, various things like that. And the other thing that, and again, one of my supervisors kept reminding me is, with your PhD, what do you want to do at the end? Like, that should inform the kind of skills and things that you want to do during your PhD. And I've kind of got a little table to the right here, kind of summarize what I thought were the three main things you'd want to get out if you went from more academic or a more industry type route. But just one word of advice: avoid doing too much. It's one thing I was quite guilty of during doing my PhD. Right, so moving on from the PhD to where I am now. So this is the previous graph showing my background. So I ended up, once I finished my PhD, I managed to get a postdoctoral scientist position, which is probably quite unusual for statisticians and for a statistician in a CRI in New Zealand. So this came about because we had another MB project that had funding for a postdoc, essentially to do more computational stuff. And then, so that was a 2-year position, and a few months ago, I ended up getting a permanent role at AgResearch, so I'm now essentially a consulting statistician. So I just want to talk a little bit about what it's like to work in CRI in that more consulting statistician role. So just a bit of background on AgResearch. So we've got four campuses across the country. We focus on research that's in agriculture for New Zealand farming industry. So it can range from the animals themselves to what the products produced from the animals, or the management of farms and its practices. It also relates to research on forages, so essentially the grass the animals eat. And there's also a lot of research in kind of environmental things as well. So the company's got around 850 staff, I think with the latest report was the number. And we have 17 statisticians in the company. That covers about 12 full-time positions, essentially. So what kind of projects do we kind of get on a day-to-day basis? So the first type of project, well, not project, but essentially a lot of the time we sometimes just have conversations with scientists or whoever it is. And it can range from a quick conversation over a cup of coffee at morning tea. And I can tell you that a lot of work often gets done over morning tea coffee, to just general advice about how to do statistical analysis and things like that. And then we kind of have smaller projects where it might be like we get a pilot trial to analyze or one-off experiment to analyze. We have animal ethics forms, and I'll talk a little bit about that later, and or experiment designs, things like that. And then we have our larger projects, which are pretty much long-term, ongoing collaborations with scientists. So such as like multi-year experiments, multi-million-dollar research projects. I've been involved in a few now, things like that. So yeah, there's different scales of projects we deal with on a day-to-day basis. And one thing I've tended to find is, particularly the smaller projects, if you do a good analysis for a scientist and give them a really nice result they're happy with, you almost guarantee they're going to come back and ask for more. And so it's almost a guarantee. Right, the other thing about working as a statistician in CRI is you often work on quite relevant, and for me it's somewhat topical research. So I've been involved a lot lately in sort of methane related research. So for those who may not be fully aware, in New Zealand there's been a big push in the last couple of years to reduce methane emissions in the livestock industry. And down the bottom is probably the key. So the current goals is to cut methane emissions by 10% in 2030, and to 24 to 47% by 2050. So as a agricultural research institute, our job is to give the farmers the tools to be able to achieve these goals. And so we've been doing quite a lot of research in this area that I've been involved in. So up top left here is a way that we're using routinely now to measure methane in sheep. So they get put in these chambers that are on the back of a trailer and get carted around to farms so we can measure sheep on farms. We have a particular sheep flock that's been breeding for the last sort of 10-15 years. and it's been bred specifically for divergent methane. And we've done a lot of trials on these animals, just seeing what effect breeding for different divergent methane on these animals actually has. And so that's been quite a successful program as well. And then probably what has really been the guts of my postdoc is looking at the rumen. So the rumen is the stomach of livestock animals that essentially is required to break down the feed, the grass the animals eat. And so whether the information sort of in the microbes in their stomach, whether that can be used to predict or inform whether an animal is going to be a low or high emitting methane animal. So this is, yeah, to me, it's working on relevant, cutting edge, topical, whatever's going type research. So I mentioned animal ethics earlier. I didn't actually realize that there was ethics for animals until I started working ag research. I mean, universities, they talk a lot about ethics for human experiments, which I think probably a little bit more stringent. But so with animal ethics, it's a requirement by law that any experiment you do on animals, that you have an animal ethics form filled out. And these are kind of underpinned by 3 principles that essentially, can you replace animals? Can you reduce the number of animals, or can you refine your experiment? And I've got a screenshot on the right here, roughly, of what the bioinformatician has to fill out as a statistician in terms of the animal ethics, and often involves performing a power analysis and also helping co-design the experiment. And yeah, this might be a bit of a surprise to many people, but as part of my postdoc and as part of working ag research, I've done a little bit of field work. Actually, it was quite hard finding photos of me actually out in the field, but this is about the best one I've got. It's the top middle there, where we were actually doing a slaughter on the divergent methane animals. I've actually found that doing field work is extremely useful in understanding data, because understanding the errors or how the errors in your data can creep in, but also in terms of how you can help the lab team or the field staff, help the processes in terms of making the data a lot more robust and streamlined. And funny enough, I remember when I first started ag research, that I think one of the statisticians, old timers, said that they used to do a lot more field work or help out in the field back in the day. Once in a while it was. So we kind of lost it a little bit, but maybe this is coming back. And then, yeah, the data is often messy. So we get errors, sample errors. So the biggest one is always the sample IDs. Often they're handwritten at times if you're out in the field and you have a tag that isn't on your printed label, you have to write it on. Samples can go in the wrong tubes, things like that. You can end up with corrupt treatment groups. So maybe your sheep jumped the fence. That has happened. Yeah, you have intended measurements that be collected and then something happens, like animal ends up dying, or you can't get the sample out of an animal and things like that. So that all of a sudden makes your data set a lot less tidy. This one happens far too often. The data sets come in formats that are inconsistent. Your column names are different. Your header information is different and all sorts. And then, yeah, would you believe it? I've had this one as well. Come up, get asked to do an experiment. Someone says the data's around somewhere and no one actually knows where exactly it is. And then we have to go around hunting and finding the data. And it's like, particularly for a big project where there's been lots of different samples collected and processed from different people, sent to different people. Yeah, just collating that can be a real challenge. And also, while we work as a CRI, we actually still do a bit of teaching. So there's a couple of my colleagues who do lecturing. I've helped out with one lecture at Otago a year. We do training assigned us on site. We do actually do a lot of supervising, mentoring. So we have summer internships every year. We've been involved with masters projects. I'm actually planning to get involved with supervising a PhD student, postdocs. We're also involved in the research community, so journal reviewing, thesis examinations. We do quite often. There's a few of us who are editors or associate editors roles and things like that. And then involvement in professional bodies, and I know that previous statisticians, data research have been even president of the NZSA. So kind of just wrapping a little bit, I've gone a bit longer than perhaps I should have. But so is academia industry really at war? I haven't really talked too much about this, but I just think I was being a little bit over dramatic to try and get people to come along. But what I want to get out of this presentation is that really they're different, and different in their own ways and in good ways. So both have their place and purpose. I think academic has a real place in developing methods, and taking that time to make things robust, and there's a real need for that. But at the same time, industry is a lot closer to the application. It's much easier to understand what the needs are of industry. And so, and in terms of, I suppose, I'm thinking of those younger statisticians on the call here, thinking about what kind of career they might want to pick. It really comes down to what kind of person you are and what makes you tick. Like if you really like to do freedom in your research, then academic routes are much more appealing. If you want to work closely with application, or you actually want to see what you develop actually sort of implemented directly and make impact in society, then I think an industry route is much more appropriate. So with that, so where's the gap is the way I put this. It was very interesting that after this presentation was advertised, there was a bit of we got a bit of interest or feedback from various people, and in fact one of my colleagues got emailed from Gene Thompson, who I didn't actually know about before he told me about it. Used to be president of NZSA, was part of the DSIR back in the day. So the DSIR was essentially all this Crown Research Institutes in New Zealand combined together, more or less, and was split up in 1992 when AgResearch was formed. And I thought she made some interesting comments to me in the email, and saying that essentially back in the day there was the Applied Maths division. We basically, the theory and practice were being worked on together by the same person, and that this approach seems to have fallen into history of more than 30 years ago, which is essentially when the DSIR dissipated. So I mean, it might be a little bit extreme, but perhaps there's a point to this. Yep. And I think it really comes down to funding models. And in the CRIs now, it's very hard to get funding to do statistical method development all the time, for a matter of fact. The only way to really do it is to be involved in a wider research program where the methods you develop are actually providing the solution to the program to answer the biological question. And it's not always easy to find scope for those sort of things. So I just wanted to finish off with where I think academia industry could perhaps work together a little bit more closely. And I actually think both parties probably need to do a little bit better at this. So if we think about academia, what are these strengths? They have mathematical, they have the expertise, particularly the mathematical expertise, I think. They've got postgraduate, undergraduate students, and they've got probably more time and funding for method development. But I suppose the limitation I've seen with academia is that getting access to data sets isn't always easy, particularly real life data sets. Whereas in industry, we're much closer to the application. There's a lot of statistical and biological expertise that's readily available to us. We occasionally have interns, but it's quite hard at times to get PhD students in statistics, that is. We have a lot of PhD students in other areas, but actually in statistics, all that sort of specialty is quite difficult. And most of the time we're just too busy trying to keep up the day-to-day tasks to actually be able to do method development, or even publish it for that matter. But we have lots and lots of data sets, more data sets that we have time to actually analyze. So if you combine probably the good things from both sides, I think there's a real opportunity to be able to really develop statistical methods, or to have statistical method development that is informed by the application. And yeah, that's kind of, yeah, what I want to say, and probably the big thing I wanted to get out of this. All right, so acknowledgements. I just want to acknowledge all the people who've really played a key part in my development as a statistician over the years. Many of these are my supervisors. So I suppose Professor Martin Hazelton is my undergraduate supervisor at Massey when I was there. Professor Alan Welsh when I was in Australia. Matt Schofield, Mick Black, Phil Wilcox, Ken. When I was doing my PhD. Suzanne Rowe was essentially my mentor for my postdoc, and all the various other people, students, academic staff, and science staff, etc. And then if you look at the last name here, this person, there is a connection. She's actually my mum, and she is probably the main reason why I ended up getting into statistics, because she was studying statistics when I was growing up, or retraining statistics, ended up doing a PhD, finished a few years before I did. And I got to see sort of the cool things that you do with statistics. So credit goes to her. And finally, if you just want to contact me, any questions or just want to yarn, you can easily email me, so timothy.bilton@agresearch.co.nz. Probably the best way to get hold of me, though I do have a few social media platforms. So with that, I'd like to wrap it up, and thank you all for listening.